Later on, the experiments of the grip were continued by 3 people, that is, Avery, MacLeod, and Cott. What they have taken is the final step of grip experiment. That is, in mice they introduce, that is, the final step of grip experiment is the S strain, heat killed, and odd strain, which has led to mice type. What is the reason for this? So they tried to find out what is the substance that is transferred, what is the substance that has caused the death of mice. So for this, what they have taken is, along with S strain, heat kill S strain and odd strain, they have kept, that is, they have, along with, they have kept proteases. They injected proteases. Proteases are enzymes which will digest proteins, but the same final output, that is mice dies. That means proteins are not responsible. Along with the heat kill S strain and odd strain, so they introduced nuclease which digests nuclease. Here mice survives. So here it shows that nucleic acids causes transformation. That means transformation means nucleic acids are transferred from one strain to other strain. Okay, fine. Which nucleic acid, whether it is DNA or RNA. So to find it out, whether it is DNA and RNA, we should find it out. To find it out, the next step, along with heat kill S strain or strain, they introduce RNAs. RNases means which will digest RNA. But the same result, mice dies. So RNA is not responsible. In the final step, along with S strain and R strain, they introduce DNAs, where the mice survives. That means the confirmation, that is the final output of this the step is DNA is responsible for transformation. That means from heat killed S strain to R strain, the DNA is transformed. So which has led to the R strain has become into the lethal S strain. So which has caused, that is, the mice. Okay. So they have given a final thing, that is DNA is transformed. So how to confirm that whether DNA is transformed or not? When we come to the next experiment, that is Alfred Hershey and Martha Chase. So they have conducted experiments on bacteriophages. Bacteriophages means virus that infects bacteria. So viruses that infect bacteria, that is called bacteriophages, which has DNA as a genetic material. So in bacteriophages, they have taken bacteriophage T2, which contains double stranded DNA. And when you come to bacteriophage T2, that is, when you take virus, it is made up of protein coat. Bacteriophage T2, it is made up of protein coat and DNA. To carry out this experiment, along with bacteriophage T2, they have taken radioactive isotopes, which can be used for tracing. So they have taken 2 radioactive isotopes, 2 separate experiments were conducted. What is that? We'll see. So here what they have taken is, that is, one is they have taken one radioactive isotope, that is phosphate, which is present in DNA. Which is present in DNA. Phosphate is present in DNA. And they have taken radioactive sulfur, which is present in proteins, but not in DNA. So sulfur is present in proteins, whereas phosphate is present in DNA. To have a clear differentiation and a confirmed output, they have taken radioactive phosphate and radioactive sulfur. That is, to get the T2 bacteriophages, first viruses are grown in medium containing phosphorus 32. In other experiment too, they have grown viruses, that is T2 bacteriophage in a isotope 35S. So when they are grown in medium containing phosphorus 32 and sulfur 35, they have attained the labeling, that is phosphorus 32 and phosphorus 35. That is, the DNA which is present in this, it has attained the phosphorus labeling, and the protein which has here they have got, that is the sulfur labeling. That is the sulfur labeling. Okay. So here, in the step of this first step of this experiment, so they have carried out, that is, virus are allowed to infect bacteria. That is, bacteriophages will infect bacteria. After infection, so they have carried out, the next one is blending. The next step is blending. Where in blending, they use a blender, an equipment called blender, where the bacterial cells were agitated violently to separate, that is, infecting virus from the bacteria. So during the infection process, the virus will attach to bacteria. So blenders will separate from bacteria to virus. And later on, the next step they have carried out is centrifugation. So in centrifugation, so the cells are separated. When the cells, all the parts of the cells are disrupted, when the cell wall, cell membrane is disrupted. So where the lower density one gets deposited at the bottom, higher density gets deposited at the bottom, and lower density deposited at the top. So if you take the experiment of that is Hershey and Chase, in this there are 3 steps. One is infection, another one is blending, and the final step is centrifugation. So he has done experiment 1 and experiment 2. In experiment 1, they have taken bacteriophage labeled with S35. S35 means, here when you take the structure of a bacteriophage, this is protein code, which is red colored, which is protein code. That means it is labeled with sulfur. This is DNA, which is not labeled with any radioactive. In experiment 2, they have taken phosphorus 32, where only DNA is labeled with phosphorus 32, nowhere protein is labeled. Why? Because in protein there is no phosphorus. So they have taken 2 experiments, experiment 1 and 2, where in both the experiments the steps are common, where they infect the bacteria. That is, the bacteriophage will infect the bacteria. When the bacteriophage infects bacteria, whatever the DNA is present, it is passed on to the bacterial cell. So here, during this process of infection, the bacteriophage attaches to the bacteria. So after infection, it is left out, the protein coat is left out, which is called coats. It doesn't have DNA. In the second step, in blending process, the virus, that is bacteriophage, is separated from bacteria. And next they have gone for the final step, that is centrifugation. In centrifugation, the bacterial cell is ruptured, where the higher density one gets deposited at the bottom and where lower density gets at the top, that is supernatant. That is, if we take the experiment 1, in experiment 1, that is in supernatant means they will have the proteins which have the radioactivity S35. Whereas when you come to experiment 2, there is no activity in the supernet. That is, there is no proteins, doesn't have any labeling. So what it says is, in this experiment, what it says is, during the process of infection, whatever the DNA is present in bacteriophage, it is transferred to, that is, the bacterial cell. It is transferred to bacterial cell. So this experiment has finally gave a confirmation, that is, it has given a confirmation, that is, DNA is a genetic material. Okay. After Griffith experiment and Avery, MacLeod and Corti experiment, Hershey and Chase experiment has given a confirmation that DNA is a genetic material. As we already discussed, what are the criteria for genetic material? When you take the criteria, it should be able to replicate on its own. That means it is able to replicate. It should be chemically and structurally stable. It should undergo mutation slowly. And it should be able to express Mendelian characters. So in these 4 criteria, if you take the proteins, they are not able to fulfill this criteria. As well as when you take RNA and DNA, they are able to fulfill the criteria. But when you take specifically whether RNA or DNA, DNA is able to fulfill the criteria. Why? Because RNA doesn't undergo replication. One RNA cannot form another one. Okay. So why DNA is taken as a genetic material, not RNA? Why DNA is stable? Why RNA is not stable? What is the reason behind this? The stability of DNA is due to the main reason it has 2 strands and it has deoxyribose sugar. Right? The OH group at carbon atom 2 in ribose sugar is replaced by only H. Due to this, it makes less reactive and more stable. And thymine gives more stability instead of uracil. That means in DNA, uracil is not present, only thymine is present, which gives more stability. So till now, what we discussed is about the structure of DNA, the set of experiments of genetic material of DNA, the criteria of genetic material, how they will decide the factors, that is, how we can decide whether it is a genetic material or DNA. Proteins are not able to fulfill the criteria, RNA is not able to fulfill, and what is the reason for DNA is able to fulfill. As it is double stranded, it is stable, and it is the next is it is very less reactive, it can undergo slow mutations, as well as it can express Mendelian characters. So that's why DNA is taken as a genetic material. Thank you all. Follow us on social media. We'll meet again in the next session.